epithelial tissue slide. This slide comes from the kidney. In the kidney we have renal corpuscles and renal tubules. That allows us to recognize the organ as the kidney. This is a renal corpuscle. This is another renal corpuscle. The kidney tubules are, for the most part, in cross-section in this particular image. This is a cross-section through a kidney tubule. This is another kidney tubule. Here's another kidney tubule. Now, the renal corpuscle is composed of the glomerulus, and surrounding the glomerulus is Bowman's capsule. Both the glomerulus and Bowman's capsule are made of simple squamous epithelial tissue. The kidney tubules, such as this one, are made of simple cuboidal epithelial tissue. Now we'll increase the magnification and we'll be able to see all of this in a little more detail. This is high magnification, looking at a renal corpuscle. This is the glomerulus. Again, that's made of simple squamous epithelial tissue. This is Bowman's capsule, also made of simple squamous epithelial tissue. Over here to the right, we see parts of a couple of kidney tubules. Here's part of another kidney tubule. Now let's focus on a kidney tubule and see if we can see the nature of the cells that form the kidney tubules. This is a high magnification view showing some kidney tubules. This is the lumen of one tubule. This is the wall. Simple cuboidal epithelial tissue forms these kidney tubules. Here's another kidney tubule. Here's another kidney tubule. And if you look right here, for example, you can see the cubed shape of the cells that form the kidney tubules, hence the name simple cuboidal epithelial tissue. So in the kidney, we have two sources of tissue that are very, very prominent: simple squamous epithelial tissue and simple cuboidal epithelial tissue. In the lab, we have another source of simple squamous epithelial tissue to look at, so let's go to that slide now. This is our second slide of epithelial tissues, and it's also our second source of simple squamous epithelial tissue. This slide is taken from the lung. This is low magnification, and what we see are numerous alveoli. This is one alveolus. This is another alveolus. This is an alveolus down here. The tissue forming the alveoli is simple squamous epithelial tissue. This tissue is specialized, in this case, for gas exchange. The alveoli are the microscopic structures in the lung where gas exchange occurs, gas being exchanged between the lung and the blood, or between the alveoli and the blood. Now this is low magnification. Let's go to high magnification. This is high magnification of an alveolus in the lung. This is one alveolus right here. While I honestly believe that it's easier to recognize lung as lung with low magnification because you have more in your field of view, I wanted to show you an alveolus at high magnification to point something out, because there are a few tissues that you might initially think look very similar. But when you look at the alveolar wall, you can tell that it is cellular. You can actually pick out nuclei. And right here, I can make another point. See, there's a nucleus here, and just to the right, there's another nucleus. In other words, while this is simple squamous epithelial tissue, the thing to keep in mind is that there are actually two alveolar walls here. There's an alveolar wall belonging to this alveolus, and an alveolar wall belonging to this alveolus. So as thin as this stuff is, it's actually two cell layers thick, one cell layer per alveoli, or per alveolus, but we have two alveoli sandwiched up against each other. So we've seen two sources of simple squamous epithelial tissue. We've seen it in the kidney forming the glomerulus and Bowman's capsule, and we've seen it in the lung forming the alveoli. We've also seen our first source of simple cuboidal epithelial tissue, in the kidney, forming the kidney tubules, where it is specialized for selective reabsorption. Now let's go to the thyroid gland, where we will see a second source of simple cuboidal epithelial tissue. This slide is taken from the thyroid gland. This is low magnification of the thyroid gland. And what we're looking at are follicles in the thyroid gland. So this is a follicle, this is another follicle, another follicle, another follicle, and so on. The walls of these follicles are made of simple cuboidal epithelial tissue. In other words, this area here is made of simple cuboidal epithelial tissue. Now, while I don't think it's a good idea to use color by itself to recognize the different tissues or the different organs, the thyroid gland is one where you're relatively safe doing that. And that is because these thyroid follicles are producing the thyroid hormone, and they stain this color. In other words, it's a mixture of material known as colloid, and the colloid stains this color. Now let's increase the magnification and see if we can see the nature of these cuboidal cells that form the thyroid follicles. This is a high magnification view of the thyroid gland. We have a couple of follicles in our field of view. This is a thyroid follicle. This is another one. Here we can get a sense that it is one layer of cubed-shaped cells. But we can also see something else. If you look at the cells right through here, they do look fairly cubed. Compare that to these cells. There's a layer of cells here belonging to this follicle, and there's a layer of cells here belonging to this follicle. So actually, this layer as well as this layer look pretty flat. They look more squamous. In the thyroid gland, when you're looking at the follicles, the terminology is low cuboidal and high cuboidal. The cuboidal cells that look squamous are called cuboidal, but the convention is to call it low cuboidal. So there's a layer of low cuboidal cells here forming this follicle, and a layer of low cuboidal cells here forming this follicle, while up here, this layer of cuboidal cells that belongs to this follicle would be called high cuboidal. What happens is, if the cells are actively producing thyroid hormone, they get thicker and are called high cuboidal. When they're sort of inactive and look more squamous, they're called low cuboidal. The third type of tissue we're going to look at comes from the small intestine, and it will be simple columnar epithelial tissue. This slide is taken from the small intestine, and it provides us with an example of our third source of epithelial tissue, that is, simple columnar epithelial tissue. The structure that you're looking at, or actually you see parts of several of them, but let's focus on this one for a moment. This is a villus. The small intestine is convoluted into villi. I'm going to move the slide over a little bit. So now you see just bits and pieces of the villi. This is the lumen, the center of the lumen of the small intestine. And so whether we go left, right, up, or down, we'll see the villi. They are simply these finger-like projections. The lining of the small intestine is convoluted into these villi to increase the surface area. The lining of the small intestine is called the mucosa. And this mucosa is formed from one layer of columnar-shaped cells. So this is simple columnar epithelial tissue forming the mucosa of the small intestine. These dark spots that you see are the nuclei. The basement membrane of this simple columnar on this side would be here, and on the opposite side would be here. The tissue in between the two basement membranes is called lamina propria. The lamina propria contains blood vessels, both to oxygenate and otherwise nourish the tissue, as well as to absorb nutrients, the products of digestion. There's another structure in there called a lacteal, which is important in the absorption of the products of fat digestion. All of this will be covered later, actually in the second semester of Human Anatomy and Physiology, when we talk about digestion. Let's zoom in on the cells of a villus. This is high magnification, looking at the small intestine. This is one villus. Moving out this way. We're getting into the lumen of the small intestine. But if you look, you can actually see that these cells are columnar. In other words, free surface is here, bound surface is here, and the cells are not very wide. They're shaped like columns or rectangles. We see another very prominent type of cell in the simple columnar of the small intestine, and here are several of them. This is a goblet cell. This is another goblet cell. Now, goblet cells are found in many types of epithelial tissue, and in all instances what they're doing, they secrete a protein called mucin. Mucin mixes with water and forms mucus. And again, this material within a villus is called the lamina propria. So we've seen the three types of simple epithelial tissue so far: simple squamous epithelial tissue, simple cuboidal epithelial tissue, and simple columnar epithelial tissue. Now we're going to look at a stratified epithelial tissue. This epithelial tissue is called non-keratinized stratified squamous epithelial tissue. Now, that is in contrast to the next one we'll see, which is called keratinized stratified squamous epithelial tissue. In both cases, the tissues are stratified, that is, more than one layer, or more precisely, to be stratified means that not every cell in the epithelial tissue reaches the basement membrane. The difference between keratinized and non-keratinized is that keratinized stratified squamous has the protein keratin, which kills the cells. In non-keratinized stratified squamous, the protein keratin is absent. Keratin dehydrates tissue, and the result of dehydrating or drying out the tissue is that the tissue dies. So this is non-keratinized stratified squamous, and you'll find that they're very easy to tell apart. Now, this is low magnification. The basement membrane would be right along here. The free surface of the epithelium is here. Stratified epithelial tissue is specialized to protect against abrasion. I'm going to increase the magnification now. This is a high magnification view of non-keratinized stratified squamous epithelial tissue. And as I was saying, stratified squamous epithelial tissue is specialized to protect against abrasion. And because this type is non-keratinized, it's also important in maintaining a moist environment. With stratified tissues, the shape is derived from the uppermost cells. So although these deeper cells may appear more cuboidal, as you work your way up, you can see that the cells are becoming flatter. And so regardless of the shape of the cells down deep, the uppermost cells are squamous, hence the name stratified squamous. Now, I can tell that this is non-keratinized because I can see nuclei throughout the stratified tissue. Even way up here, I can see nuclei in the cells. When we compare this to the keratinized stratified squamous, you're going to see a world of difference because the protein keratin dehydrates the tissue, that is, dries it out, and it subsequently dies. The cells lose their nucleus. In other words, they're anucleated. Very often in a non-keratinized, even one of these cells that's sloughing off, like this one, you'll be able to pick out the nucleus. But when we look at the keratinized stratified squamous, it will look more like fish scales than tissue. So let's go to the keratinized stratified squamous epithelial tissue now. Try to hold on to this image to compare. This is keratinized stratified squamous epithelial tissue. This comes from the skin. Now, the non-keratinized is found in places such as lining the mouth, lining the esophagus, lining the vagina. But the keratinized stratified squamous is found on the skin, specifically the epidermis of the skin. The basement membrane of the keratinized stratified squamous epithelial tissue is along here. And so everything above the area I'm indicating is the epidermis of the skin. The free surface of the epithelial tissue is up here, and actually it may be a little above that. Okay, so the epidermis extends all the way up to here. This doesn't even look cellular, does it? Now, this is low magnification. We'll increase the magnification in just a moment. But even at this low magnification, you can kind of get a sense that these cells are more cube-shaped. And as we go up, the cells are becoming flatter. And you get up here and it just looks like something dry, more like fish scales. Let's increase the magnification. This is a high magnification view of part of the skin, the basement membrane of the epidermis. And again, it's the epidermis that is formed of keratinized stratified squamous epithelial tissue. That basement membrane would be along here. You can definitely tell now that these deeper cells are more cubed. As we work our way up, watch what happens to the shape of the cells. They're flattening, they're dying, they're losing their nucleus. And we get up here into the region of truly keratinized tissue, and it doesn't even look like tissue at all. It just looks like fish scales or something. I'm going to go back to low magnification for a moment. Okay, I've gone back to low magnification, still looking at the skin. It's a good place to make an important point about these slides, just to get our bearings. Here's the interface right here between the overlying epidermis, made of keratinized stratified squamous, and the underlying dermis. And you see how the epidermis and dermis go together kind of like interlocking fingers. But people, these tissues all come from organs. And so this is a slide of the skin. As such, we can use this slide to see not only keratinized stratified squamous epithelial tissue because it forms the epidermis of the skin, but we can use this same slide, and in fact, your notes and supplemental documents will refer you to this same slide for one of the connective tissues. This interface that I've mentioned is actually between the epidermis and the dermis, or between the overlying epithelial tissue and the underlying connective tissue. When we get down here, we're in the dermis of the skin. And the dermis of the skin is composed of dense, irregular connective tissue. As we go deeper, we get into another type of connective tissue. This is adipose connective tissue. This is fat. And so you can see all three of those types of tissue on this one slide. As you're studying the slides in the lab, when you find yourself recognizing a tissue that is not even referenced on the slide that you're looking at, that'll be a good indication for you that you're really starting to recognize these tissues. Let's go to our next type of epithelial tissue. This is our next type of epithelial tissue. This tissue is called simple ciliated columnar epithelial tissue, and it's found in the fallopian tubes. Now, the epithelial tissue is right here. Free surface is here. Bound surface is here. This is the part of the lumen of the fallopian tube. So our epithelial tissue runs along here, forming the lining, and we know that epithelial tissues form linings and coverings, forming the lining of the fallopian tube. Now, this is called simple ciliated columnar epithelial tissue. So the cells are going to be columnar or rectangular in shape, one layer thick, and they're going to have cilia on their free surface. Cilia are not going to be terribly easy to see, but I believe we'll be able to see them. Let's go to high magnification and have a look. This is a high magnification view of simple ciliated columnar epithelial tissue in the fallopian tube. This is part of the lumen of the fallopian tube. This is the epithelial tissue that we're looking at. This is the free surface, and you can make cilia out right here. This is the bound surface. In fact, at this magnification, you can even tell that these cells, such as this one, are longer than they are wide. They are columnar in shape. You can pick out the nuclei of the cells. I'm going to attempt to make a minor focus adjustment here. Now, while these look like cilia, these don't. What I see here, it looks to me just like a rough surface. And quite often when you're looking at the fallopian tube, the cilia do not stand out. What you get is simply something that appears as though the free surface of the epithelial tissue is rough rather than smooth. These are fairly obvious along here. This is another area in the same fallopian tube. If you look along here, along this free surface, you can kind of get a sense of cilia, but not terribly obvious. Same thing over here. Not terribly obvious. In contrast, here is an area in the same fallopian tube where the cilia are very, very obvious. So to be honest with you, simple ciliated columnar epithelial tissue and your slide of the fallopian tube is one that you're going to spend a little time with. It's going to be a little more difficult to recognize initially than some of the others that we've seen. There's another type of ciliated epithelial tissue. Let's have a look at it now. This slide comes from the trachea, and we're using this slide right now to look at pseudostratified ciliated columnar epithelial tissue. But before we talk about pseudostratified ciliated columnar epithelial tissue, let's talk about how you can tell that this is a slide of the trachea. And again, let's keep in mind that these slides come from organs, and organs are composed of two or more tissues. This tissue right here is called hyaline cartilage connective tissue. It's one of three types of cartilage, one type of connective tissue that we'll study. The hyaline cartilage is forming the rings of the trachea, which are there to keep it from collapsing. This is the pseudostratified ciliated columnar epithelial tissue. So I can tell that this is the trachea because I see the pseudostratified ciliated columnar epithelial tissue, and I know that's a mouthful to say, but I see that, and I also see hyaline cartilage. So when you see these two together, you know you're looking at the trachea, or at least some part of the upper respiratory tract. Now let's increase the magnification, and we're going to focus on this tissue, which is the pseudostratified ciliated columnar epithelial tissue. This is a high magnification view of pseudostratified ciliated columnar epithelial tissue from the trachea. The free surface of the pseudostratified ciliated columnar epithelial tissue is here. The bound surface is here. This is actually the basement membrane along here. Pseudostratified ciliated columnar epithelial tissue is one of the few types of epithelial tissues where you can actually pick out the basement membrane with the light microscope. Now the cilia are pretty prominent, pretty obvious through here. This would be the lumen of the trachea. Let's just move the slide around a little bit, keeping our eye on that epithelial lining. Okay, so this is it. The pseudostratified is here. And you know, I'm very sure that I see nuclei at different heights, or nuclei stacked on top of each other. this tissue definitely looks stratified, but it's not. And that's why it's called pseudostratified. It appears to be stratified, but it is in fact simple. Remember that for a tissue to be stratified, some cells don't contact the basement membrane. If in a simple tissue, every cell contacts the basement membrane, but in a stratified tissue, there are some cells that do not contact the basement membrane. And what happens in this, the pseudostratified tissue, is you actually have three types of cells. There are basal cells, which are very, very short cells. These cells that have their nucleus down low are most likely basal cells. So they're very, very short cells. They're not even columnar, but they do contact the basement membrane. They are mitotically active. They give rise to the other types of cells. Another type of cell that you have in pseudostratified ciliated columnar epithelial tissue are goblet cells. And again, as I mentioned when we were looking at the small intestine, the goblet cells secrete a protein called mucin, which combines with water to form mucus. And then the third type of cell that you have here are the actual ciliated columnar cells. So because of the three types of cells, and even though all three types of cells, even though every cell contacts the basement membrane, because of the three types, and because those basal cells are short, we get the appearance, and it's not just an appearance, it's quite real, of seeing nuclei stacked on top of each other. And so the tissue appears stratified. This is a great example of where we have to trust the people who have studied the tissues with the electron microscope. And we know from electron microscopic studies that the tissue is, in fact, simple. It is not stratified, hence the name pseudostratified. One last look at these cilia. Let's recall how we know that this is the trachea. Because in addition to the pseudostratified ciliated columnar epithelial tissue, we have hyaline cartilage. So this is hyaline cartilage, in this case forming the rings of the trachea. Hyaline cartilage also forms the fetal skeleton. It's also on the ends of long bones to provide a smooth surface for articulation. It also forms a costal cartilage where your ribs articulate with your sternum. We'll talk about hyaline cartilage in a little while in the context of structural connective tissues. First of all, though, let's finish the epithelial tissues. We have two more to go. This is called transitional epithelial tissue. We're looking at a cross-section through the ureter. The lumen of the ureter is in here. And so this is the epithelial lining. It is a stratified tissue, but the word stratified is not in the name of the tissue. The tissue is called transitional epithelial tissue. The name is because the shape of the tissue changes. That is, the shape of the individual cells undergoes a transition. This is actually easiest to recognize on low magnification where you get the big picture, and a lot of people think that this just looks like a star. If we increase the magnification, we'll get a little better sense of the nature of the transitional epithelial tissue. But when the ureter or the urinary bladder, and those are the two locations of transitional epithelial tissue, when they are empty, the tissue is sort of oval, sort of egg-shaped, something like that. On the other hand, when the urinary bladder is filled with urine, and it stretches, the tissue stretches out, and so the cells go from sort of a almost cuboidal, but more of an oval or egg shape, to actually very, very flat, very squamous looking. Let's increase the magnification. Okay, this is a high magnification, a part of the ureter. This would be the lumen. So this is the free surface of the transitional epithelial tissue. The balanced surface is along here. So you can see it's definitely a stratified tissue, and the cells are sort of round, sort of oval, sort of egg-shaped. And just try to imagine that if this was filled with urine and everything was stretched out, the cells would be very, very flat. Hopefully you've seen the video in the library called epithelial tissue, and Dave shows you a picture of both a full and an empty urinary bladder, making the point of how the tissue undergoes the transition. We have one more type of epithelial tissue. This slide is taken from the pancreas. The pancreas does not fit the nomenclature of epithelial tissue. It actually belongs to an epithelial category known as glandular epithelium. The group glandular epithelial tissue would include all of the glands, and if this was a class in histology as opposed to a chapter in a textbook, we would cover most, if not all, of the glands. But I like to use the pancreas as an example of glandular epithelial tissue because it's very interesting. It's very interesting because it belongs to two distinct systems of the body. The pancreas is an exocrine gland, meaning that it is a gland that has a duct, and insofar as its exocrine gland activity, the pancreas functions in the digestive system. It produces digestive enzymes. They travel through the pancreatic duct and are released in the small intestine, where they function in the chemical digestion of food. The pancreas also functions in the endocrine system. Endocrine glands are glands that don't have ducts. Endocrine glands produce and release hormones into the blood. Insofar as its endocrine gland activity, the pancreas produces insulin and glucagon. Insulin is used to promote the uptake of glucose, meaning move the glucose from the blood into the tissue. And glucagon promotes the hydrolysis of glycogen, that is glycogenolysis, which results in releasing glucose from the tissue into the blood, raising the blood glucose level. So overall, insulin will lower your blood glucose level. Glucagon will raise your blood glucose level. But the types of cells in the pancreas that participate in these two different systems are quite distinct. Now, this is high magnification of the pancreas, and it's just exactly where it needs to be. This group of cells in the center that is lighter, let's say pink, as opposed to these which are more purple. The cells in the center form what is called islet of Langerhans. The islets of Langerhans are the endocrine system cells. So, islet of Langerhans here. These are the endocrine system cells. That is, these are the cells that produce insulin and glucagon. All of the other cells in this field of view are called acinar cells. They're the cells of the digestive system that produce the digestive enzymes. So the pancreas is unique. It doesn't really look like anything else that we've looked at. And the islets of Langerhans are quite distinct from the acinar cells.